Aurore Cournoyer is at Laval University in Canada, and she's going to speak on impacts of discoloration on the antibacterial and antifungal bioactivities of a porcine cruor hydrolysate. And I must confess, Aurora, I had never thought about the question of cruor and the fact that it's a rich raw material and a source for a lot of potential products. So you started in September 2020 at Laval, and so you are just really launching into the hard work of it now. So I'm very excited to hear your paper. If you wouldn't mind turning off or turning on your microphone, and I will be glad to hear what you have to say. Thank you.
Okay. So hello everyone. So I'm going to present you today a part of my PhD thesis, which focuses on the in vitro antibacterial and antifungal bioactivities of a porcine cruor hydrolysate. So first, it's important to know that the pork industry is one of the main sectors in the agri-food field in Québec. They provide about 25 to 30% of the Canadian consumption of pork. Nevertheless, consumers' concerns regarding this sector are increasing. They particularly care about the pollution, the production of waste, and the use of synthetic preservatives. And slaughterhouses produce many byproducts such as blood, which represent 30 million liters per year in Quebec. So you have to know that the blood can be separated in two parts, the plasma and the cruor. And the cruor, it's the red cells part. And it contains about 90% of hemoglobin. And so thanks to its abundance in protein, it's an ideal substrate for proteolysis. And it had been demonstrated with the bovine hemoglobin that after enzymatic hydrolysis, you can produce the bioactive peptides, specifically antimicrobial peptides. And these peptides can be used in the pork industry, which creates a circular economy. But there is a lack of knowledge concerning the bioactive peptides from the porcine cruor hydrolysate. And moreover, it's important to know that the color and the metallic flavor of M that is contained in the hemoglobin can limit its commercial potential. So that's why the main objective of this study is to evaluate the impact of the discoloration of a porcine cruor hydrolysate on the antimicrobial activities and on the peptide profiles that are generated. So concerning the method, first we had to produce the cruor using centrifugation, and then we did an hydrolysis with pepsine using these parameters, and a discoloration using acidification. And then we evaluated the antimicrobial activities using a qualitative method so based on an agar diffusion, and a quantitative method which determined the minimum inhibitory concentration. So we did these both methods on bacteria, yeast, and molds. And then we also determined the peptide profiles with UPLC coupled with MSMS system, which allow to separate, identify, and characterize the active peptides. So concerning the results, we can easily see on this overlay of the raw and discolored hydrolysate spectra that there are differences between the both hydrolysates, and mainly at the end of the spectra between 20 and 27 minutes of retention time. And also you can see that no peak of M was observed on the discolored hydrolysate spectrum, which shows the effectiveness of the discolored step. So we identified the lost peptides, and then it was relevant to see whether these lost peptides had an impact on the bioactivities or not. And as you can see, no inhibition was observed on bacteria, but antifungal activities was observed for both hydrolysates, so the raw and the discolored hydrolysates. And we observed a significant differences between our two hydrolysates. So the raw hydrolysate was, for example, for the first strain, which is Saccharomyces boulardii, you can see that the raw hydrolysate is 10 times more efficient. So the strain is more sensitive to this hydrolysate and less sensitive to the one discolored. So following these results, we were able to demonstrate for the first time that porcine cruor hydrolysate have an antifungal activity, and that the discoloration causes the complete loss or the diminution of some specific peptides that have an impact on the resulting activity of the hydrolysate. So the general perspectives of this project would be to produce a new and bio-based ingredient. And also we decided to synthesize some of the lost peptides to see their activity purified, and the results are yet very encouraging. So thank you for listening, and I'll be happy to answer your questions.
Thank you, Aurora. That was very concise, and it's obvious you've gotten a great start. So I appreciate your perspective.
Yeah, thank you. Thank you very much.
Susanna, did you have a question? I see you're unmuted.
Yeah, thank you. Congratulations, Aurore. It's a very, very nice presentation, and I know nothing about your subject, but with your presentation, it was much clearer for me now. But I have some questions that are more curiosity, because I don't work in your field. And you said that these peptides can be used as food preservatives. Are they already used in the food industry, or is it new, it's idea to use them in the future? And do you think, just two questions in one, and what do you think about discoloration of your raw material, because you observe the loss of activity? What do you think about all the process in terms of use in food technology, for instance?
Yeah, so they are not yet used in the industry, but there are several studies that show that they can have very good activities in situ. They tested some peptides on the meat, and it shows a very good result. So it has the potential to be used as preservatives. And so it's not yet used, but it's a project, and it's very interesting because it could create a circular economy, and I think it's very interesting. But yeah, of course, the discoloration, it's having a new step, so it would be better not to have this step. But because of the color, if you want to add this hydrolysate on light meat or something like that, it could create an issue. So that's why we try to discolor to see if it has an impact or not. And also for later, we will try to fractionate the hydrolysate with some techniques that use membranes, and the M can be a problem. So that's why we really wanted to, at first, compare the two hydrolysates.
Thank you.
I don't know if it's answered your question.
Scott, if I may ask the next question.
Please, Professor.
All right. Thank you, sir. Appreciate it. A gentleman and a scholar you are. Scott and I are friends, so we just ignore that. All right, very good presentation. I was very interested because I'm working with antimicrobial peptides that actually attack bacteria. They're designed to, they penetrate the biomembranes and kind of open up pores. I was curious, what is the underlying mechanism, or do you know this, for the products that you're producing and how they may be affecting yeast and fungi and causing that antimicrobial activity?
But the antifungal mechanism are quite close to the antibacterial mechanism. They also have to penetrate the cell membranes, and I know fungus have another membrane that bacteria doesn't have, but it's quite the same mechanism, and the peptides will penetrate and cause the loss of metabolism, the metabolites, and then the cell will die, like bacteria.
Thank you.
Thank you.
You're welcome.
Very good presentation. In the interest of time, I'm going to skip the remaining questions, but thank you very much for a very interesting and well put together talk and a very good presentation.
Thank you.
So thank you.